Okay, we're going to continue with activity 3 now. I'm going to go ahead and share my screen real quick. And there was one thing I actually wanted to go back to. I think I didn't do a great job about explaining this. So I told you guys that a water molecule is composed of 2 hydrogens and 1 oxygen, and that these atoms form covalent bonds with each other. And this is an example of a polar covalent bond due to the electronegativity of the oxygen. I told you that oxygen has a very high electronegativity value. But then I think I kind of could have been a little bit clearer here. So as you can see here, we have a water molecule. It is formed by 2 hydrogens and an oxygen. And this is an example of a polar covalent bond. This hydrogen molecule or this water molecule also an example of a polar covalent bond. When these 2 water molecules interact, they form a hydrogen bond between each other. So I think I kind of overlooked that distinction, and I just want to make sure to point out that a water molecule is a polar covalent bond, forms polar covalent bonds. However, 2 different water molecules will form hydrogen bonds between each other. And that hydrogen bond is formed between the oxygen of one water molecule and the hydrogen of another water molecule. So I just want to make that distinction a little bit clearer. So now we're going to go on to activity 3, which talks about carbohydrates. This is the first macromolecule we will be discussing. Carbohydrates are a class of molecules that includes sugar and polymers of sugars. Monosaccharides, or simple sugars, are the monomers of carbs. These monomers cannot be broken down into smaller sugars. So examples of these monomers or monosaccharides would be glucose, fructose, and galactose. So here I have shown the molecular formula for glucose and fructose, which is C6H12O6. It is identical for both glucose and fructose. However, as you can see, the structural formula or the structural arrangement is different, and that is highlighted here by the yellow and the blue. And this would be an example of structural isomers. So these molecules have the same molecular formula, but they have different structural formulas. We will also come across a different example of an isomer, which would be the alpha and beta glucose molecules, and we will discuss this a little bit later, but it's also on page 30 of your lab manual. So monosaccharides, particularly glucose, are the #1 choice as an energy source in the cell. So your body loves to use glucose as an energy source, and it's often referred to as short-term energy storage, whereas fats or lipids are referred to as long-term energy storage. There are many enzymes in the body that are ready to break down glucose to use it. So in order to prevent this from happening, glucose can bond together to form disaccharides. So disaccharides are essentially 2 monomers that join together, and they join together through condensation or a dehydration reaction. So some examples of disaccharides would be sucrose, lactose, and maltose. So sucrose is composed of 2 monomers or monosaccharides, fructose and glucose coming together. Lactose is composed of the monosaccharides glucose and galactose, and maltose is composed of 2 alpha glucose molecules joining together. Polysaccharides are long chains of sugars. They are polymers of monosaccharides. So your monomer for a carb would be a monosaccharide. Your polymer would be a polysaccharide. An example of these would be starch, glycogen, and cellulose. So starch are long chains of glucose that store energy, commonly found in potatoes. Glycogen are long chains of glucose that store energy with some side branches. And cellulose is found in cell walls. Chitin is found in the exoskeleton of crab, and these provide structural material. So if we were in class, you would have access to different beads or balls to represent carbon, hydrogen, or oxygen, as well as different bonds, single bonds and double bonds, and you would be asked to build a glucose molecule. So we'll be doing this together. So monosaccharides are carbohydrate monomers. Glucose is the most important monosaccharide and consists of carbon, hydrogen, and oxygen atoms. Here is the molecular formula for glucose. If you wanted to do this at home, you could use a marshmallow and just color it black to represent carbon. You could cut the hydrogen or your marshmallow into 2 pieces to represent hydrogen. And you could use pink marshmallows to represent the oxygen. So using a do-it-yourself molecular model kit, we're going to build alpha glucose molecules. As I said, this is the formula. And the first thing you would want to do is begin drawing your structural arrangement, and you can follow along on page 30. So we're going to do alpha glucose. And the first thing we're going to want to do is grab our oxygen, and then we're going to grab 5 carbons and connect those with our bonds. So we'll do our oxygen and then carbon, 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 connect all those with our bonds. And you can see that done here. Now we're going to start doing the branching off of these carbons. So the first carbon has a hydrogen on top and a hydroxyl group on the bottom. So we will put a hydrogen here and our hydroxyl group here. So that is shown here, hydrogen here, hydroxyl group on the bottom. Please, please, please make sure you pay attention to this orientation. So here the hydroxyl group is on the bottom, and that is very important because we are building an alpha glucose molecule. So here we move on to our second carbon. We have hydrogen on the top, hydroxyl on the bottom. So hydrogen on top, hydroxyl on the bottom. Third carbon molecule, we have hydroxyl on the top, hydrogen on the bottom, hydroxyl on the top, hydrogen on the bottom. And then you will just follow along and do that throughout. So once you are completely done building this, you will compare your completed alpha glucose model to your beta glucose diagram. And there is actually a site you can follow to see this done in case you don't have the material at home. That is completely okay. You can follow this website and you can see an alpha glucose molecule and a beta glucose molecule completed. And essentially what you're going to see is that you could play with this and rotate it around, but essentially what you see is that they differ in the direction that the hydrogen and hydroxyl group points on carbon 1. So here's our carbon 1. In our alpha glucose molecule, we have the hydroxyl group on the bottom and our hydrogen on the top. When we look at our beta glucose molecule, at the carbon 1 point, we have our hydroxyl group on the top and our hydrogen on the bottom. So that is the main structural difference we are going to see. When you take a bunch of these alpha glucose molecules together, you can form long chains called starches. When you take beta glucose molecules together, they will form polymers of cellulose. So starches can be easily digested by animals, whereas these beta glucose molecules, when you form those together, they form cellulose, and those make up the cell walls of plants, and most animals cannot digest them. So as you can see, this structure gives very different purposes in biology. And it's such a simple difference in the structure. Just, this hydroxyl group on the bottom, this one on the top on that first carbon. So that's one example of isomers. Another example of isomers would be taking these monosaccharides, glucose, galactose, and fructose. If you were to write out how many atoms of carbon, how many atoms of hydrogen, and how many atoms of oxygen are found in each molecule of glucose, fructose, and galactose, you would see that they all have the same molecular formula, C6H12O6. So that's the molecular formula for all 3. However, when you look at the structures of these 3 monosaccharides, you see that the structures are very different. So as you can see, these are circled in red and purple for you to see these differences. So here we have our hydrogen on top, hydroxyl on the bottom. Here we have the hydroxyl group on the top, hydrogen on the bottom, and here we can see in fructose this different arrangement here. So this gives rise to very different purposes in biology. And like I mentioned in the beginning, if you were to taste glucose, fructose, and galactose, you would see that they taste very differently. So molecules of monosaccharides may have the same molecular formula, but differ in their three-dimensional structure, and this is called isomerism. So these are all isomers. So the next thing I want to talk about is how to form these disaccharides or polymers, polysaccharides. So essentially you could have a short polymer or two monomers coming together, and when they come together, this monomer will lose its hydrogen. This monomer or polymer in this example will lose its hydroxyl group. It will form a hydrogen molecule, and in the process, these two can combine now to form a longer polymer. So these monomers will come together to form a polymer through dehydration or condensation. Now, if you have a large polymer and a water gets introduced here, the breakdown of polymers occurs through hydrolysis. So now you have these two separate monomers, and we're going to do that with the structures we built. So we're going to take our 2 alpha glucose molecules, and we're going to begin with our position 1 here, our carbon 1 and our carbon 4. So our carbon 1 is going to lose a hydrogen. So we're going to lose a hydrogen here. So we're going to get rid of this hydrogen group here on our carbon 1. So this one's going to get lost. And then on our carbon 4, we're going to get rid of the hydroxyl group. So this is going to go away. So we'll go ahead and show that here. So we lost our hydrogen. This hydrogen is gone. It's floating around here, shown here. And this hydroxyl group is gone. It's floating around, and it's shown here. So now what can occur is that that hydrogen and hydroxyl group will come together and make a water molecule, which is shown here. And now these 2 alpha glucose molecules can form together, bond together, and form maltose. So here the 2 monomers have been condensed into the disaccharide maltose, and water was formed in the process. Now the reversal can happen, and this is called hydrolysis. So now this water molecule is going to get introduced to these 2 alpha glucose molecules. So it's also written out here. You have this water molecule that's going to get introduced to this maltose polymer, or disaccharide, I'm sorry. And when those get introduced, we have the reversal happen. So now we have 2 alpha glucose molecules formed. We have our 1 here and 1 here. So that's just showing how condensation and hydrolysis occur to either form a disaccharide or a polymer, or to break it apart into its smaller components. So that's it for carbohydrates. If you guys have any questions, like always, please let me know, and I'll be happy to walk you through it.